Hi everybody, so today we're going to be talking about how we taste. Okay, so let's go ahead and get started. As everybody knows, you taste with your tongue. So let's go ahead and take a look at this. And the first thing I'm going to do is I'm going to draw a tongue. This is look at the tongue from the top. Imagine somebody sticking their tongue out. Okay, so there's our tongue. Back here is going to be the posterior tongue. So this would be the part by the throat. This is going to be the tip of the tongue right here, okay? Now, if we look at the tongue, the first thing we're going to notice is we have projections on the tongue, okay? And the taste buds are inside the projections. So the first one I'm going to do is I'm going to draw these projections right here. And these are going to be called the circumvallate papillae, right? We call the projections papillae. And you have about 7 to 12 of these. So I got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. And you can see these are in a V shape. So this is my circumvallate papillae. Okay? And then after that, on the sides, we are going to have basically some folds. And you can actually see these. And these are like little folds that are on the side of the tongue. And we're going to call these foliate papillae. Okay? And then after that, we are going to have some more that are in the front of the tongue. And these we are going to call fungiform papillae. And then after that, we are going to have basically papillae that are shaped, they're actually kind of shaped like little triangles. And these are just designed to provide some roughness to the tongue so you can move food around. And we're going to call these foliate papillae. Not foliate. These are going to be filiform papillae. Okay, so those are filiform papillae. And these have no taste buds. So there's no taste buds on these, right? So that means that these do have taste buds. So let's go ahead and take a look at these. And the first thing I'm going to do is I'm going to start off with my fungiform papillae. Let me just move my camera over here a little bit. And if we take a look at this, what I'm going to do is we're going to draw this now. You're looking at the tongue from the side now. So over there was looking from the top. Now we're going to look from the side. So if I were to take one of those fungiform papillae out and then just turn it on its side, it would look something like this. Okay? And they're going to be kind of like a mushroom shape. So they're going to look something like this. Okay, if you notice we have openings up here. I only drew 2. You're actually going to have a whole lot more than 2. So we're going to call this opening a taste pore. Okay, and that's it right there. That's a taste pore right there. And then that's a taste pore right there. And then what's going to happen is coming up to this taste pore, we are going to have these little microvilli, and we call these gustatory hairs. Gustatory hairs. Now, they're not actually hair, they're microvilli, and you're going to see they actually have receptors on them. They're also known as taste hairs, okay? So then what's going to happen is we are going to have our taste bud attached to them. So here's our taste bud. Okay, and this is a taste bud also. And then coming off the taste bud, we're going to have a nerve. Okay, so here's what this is going to happen with this nerve. This nerve is actually going to meet up with nerves from cranial nerve 5, and cranial nerve 5 is going to be responsible for feeling the texture and the mechanoreceptors and all that from the tongue, okay? These are just going to be sensory and they're going to be responsible for taste. But anyways, these are going to go on and form something called the chorda tympani nerve, or that's going to be a branch of cranial nerve number 7, which we call the facial nerve. Okay, so that's cranial nerve number 7, which is the facial nerve, okay? So this is, like we said, this is going to be my fungiform papillae. Now again, the papillae is the whole projection. The papillae is the projection. The taste buds are inside of it, okay? Now, Let's take a look at the next two. So what we're going to do now is we're going to look at the circumvallate papillae, and we're going to look at the foliate papillae. So let me move this over just a little bit over here. And what's going to happen now is this. This is going to be the top here. Okay, so here's the top here, and then what's going to happen is we are going to have our pores here, okay? And then same with over here, and then we're going to have our pore. It's going to go like this, and then we're going to have another pore there, okay? And then we can just draw the bottom of this here. This is going to be the top, right? And again, we said this is going to be our circumvallate papillae and our foliate papillae. Okay, so once again, if we look on the sides here, we are going to have our gustatory hairs coming out here, right? We're going to have our taste bud, and then we're going to make a nerve. And it's going to be on both sides here, okay? And it's going to make a nerve, and then there's the taste bud. It's going to make a nerve, and then boom. Okay, now in this case, what's going to happen is this is going to be called my lingual nerve, and my lingual nerve is actually a branch of cranial nerve number 9, which is my glossopharyngeal nerve. And we're going to be talking what happens to these in a little bit, okay? So anyways, this is my taste bud again. Okay? And these are my gustatory hairs. But there's going to be more to this than this. Now I'm just going to draw half of one of these over here. So what I'm going to do is we're going to go like this, right? And then I'm going to have this, and then we're going to go like this, and this is actually going to be connected down in here. I'm going to draw more gustatory hairs, right? And then this is going to be a taste bud, and there's my nerve again. This is also going to be the cranial nerve number 9. So now what happens is this. Let's say you eat a piece of food. Let's say you eat a piece of meat, okay? So there's my meat or food, whatever it is. It can be whatever you want, candy. But what's going to happen is saliva gets around this, right? When saliva gets around it, what it's going to do is it's going to take chemicals from here, and it's going to go down into this little valley here, and it's going to stimulate these nerve receptors. When it stimulates the nerve receptors, what's going to happen is that's going to eventually send a signal to the brain. So let's go ahead now and take a look at these taste buds here real quick. So I'm going to move this camera back over here, and we're going to erase this guy that's right here. So let's erase him real quick. And what's going to happen now is if we draw my taste bud, the first thing that's going to have is we're going to have something called the supporting cell. Okay, so this is going to be called my supporting cell. Okay, and then what's going to happen is I am going to have something that's called a gustatory cell. So this is my gustatory cell right here. Okay, and this is actually what's going to be doing the sensing of the food, right? Supporting cells contain something called a basal cell, which is actually going to be responsible for growth of the taste bud. And then we're going to have this here. This is another supporting cell. And these are actually going to be kind of shaped like a flask. If you use your imagination a little bit, I imagine you could say it looks almost like a beach ball to a certain extent, you know? And so that's going to be another gustatory cell. Now, technically, you have about 50 to 150 gustatory cells in a taste bud, okay? I drew 2. The orange is going to be my gustatory cell. So, once again, this is going to be my gustatory cell. Right, it's going to be this right here, okay? Now, once again, we have the microvilli that come out from the front, the gustatory hairs that come out from the front. I drew 3, you have more than 3 on there, okay? But that's going to be my gustatory hairs. So let's talk about what's going to happen now. Oh, back in here also, we're going to have a nerve, a dendrite with a receptor. So let's talk about what's going to happen here. We can sense about 5 different types of taste, okay? The first one is going to be salt. And salt is obviously going to be from sodium, and as most people know, you get that from putting salt on food, right? The second one's going to be sour. Now, sour is due to the fact that the food is releasing hydrogen ions. And remember, if you're releasing hydrogen ions, that is basically going to be an acid, right? And then we're going to have sweet, which you guys know sweets, right? Sugars, things like that, alcohol. All right, that's going to be my sweet sensation. Umami, right? Umami is going to basically be, they describe it like a beef broth. I've heard it described as like a Parmesan cheese or an aged cheese. I'm just going to say a cheesy sensation, right, or a cheesy taste. And then the other thing too is this is very sensitive to L-glutamate. And in case you don't know, L-glutamate is found in MSG, and MSG is found in Chinese food, right? A lot of times they put in Chinese food, and that's what makes Chinese food taste so good. By the way, umami actually means delicious in Japanese. And then you have bitter. And bitter can be sensed as two different ways. It can be an alkaloid, all right? And an alkaloid such as caffeine or nicotine, right? Or it can be non-alkaloid. And if it's a non-alkaloid, that would be like if you ever put an aspirin on your tongue. A lot of times bitter will actually sense for poisons and things like that. So let's go ahead and take a look at this. Now, here's the thing. Each type of gustatory cell can only sense one of these tastes. But what I'm going to do is I'm going to make it look like it can do more than that, okay? So what I'm going to do is I'm going to take one of these microvilli here, and I'm going to blow it up and make it large. So I'm going to blow it up and make it look kind of like this, okay? We're going to make it look kind of like that. And then what's going to happen is salt and sour kind of work the same way. So this is going to be the tip of my microvilli there. And what I'm going to do is I am going to put an ion channel here. And for the most part, this ion channel is going to be called an epithelial sodium channel. So it's an epithelial cell. And knowing what's going to happen is sodium's going to drain into this or go through this to come inside the cell, okay? This doesn't really play a role in action potentials. But here's what's going to happen is as I eat something salty, I increase the amount of salt. So as I increase the amount of salt, what's going to happen with this channel? It is going to increase the amount of salt going through. So now I'm going to start getting more and more salt going through, or more sodium going through, right? As I get more sodium going through, if you remember, the inside of a cell is negative. But as I get more sodium coming through, it starts to become more positive. It's going to be called a graded action potential. So I'm going to increase the amount of sodium. And when I increase the amount of sodium, that's eventually going to lead to an action potential, a graded action potential. But the cell is going to depolarize. So that's what's going to happen here. Now with sour, it's going to be a little different. They're not quite sure of how this works. But with sour, there's two things that can happen. One is, again, I have these ion channels that are on the cell membrane, right? This black line's my cell membrane. And what's going to happen is my hydrogen ion can come down and bind to this ion channel. And when it does, what's going to happen, this should look familiar, is I'm going to start to get more sodium coming into the cell. Okay, so my hydrogen is binded onto there. This keeps that channel open. More sodium comes in. And as we saw, when more sodium comes in, I get an action potential. That's one way this can happen. The second thing that can happen is I could actually have another channel down in here. And for the most part, this channel here, potassium goes out. Potassium goes out through this channel, okay? But what might happen is when I get an increase in the hydrogen ions, right? If I get an increase in the hydrogen ions, they start to come in through this very same channel. And because they're coming in, two things are going to happen. One, I'm going to get an increase in hydrogen ions, which have a positive charge. And two, I am going to get an increase in this potassium because of the fact that it's not leaking out. So I'm going to increase the amount of potassium inside. So if that happens, it's basically going to be the same thing as here. It's going to lead to the action potential, right? Another graded action potential because of the fact that I'm getting more positive charges on the inside. So now we've depolarized our cells. So let's talk about what's going to happen next. Once we do that, you're going to have down here, you are going to have a calcium channel. Okay, these are going to be calcium channels. So what's going to happen now is calcium's going to start rushing into the cell. So we depolarize the membrane, and now calcium starts rushing inside. Okay, and here's another one, right? And calcium's going to start coming in. So what happens if calcium comes in? Well, inside of here, we are going to have a neurotransmitter that I'm going to call serotonin, right? And serotonin is going to be inside something we call a vesicle, right? So this is going to be a vesicle. And then my vesicle is actually going to have some proteins attached to it. So here's the proteins, right? Those are proteins, right? And then what's going to happen is I'm going to have my cell membrane over here, right? Here's my cell membrane. And my cell membrane is also going to have some proteins attached, right? So when calcium comes in, what calcium's going to do is it's going to connect these proteins, right? And when it connects those proteins, what's going to happen next is it's going to pull this vesicle, which is made up of the same thing as the cell membrane, and it's going to release the serotonin. So I'm just going to draw it like this, okay? And then let's go like this. I'm going to draw this. Let's just say this is my serotonin, right? And now these are going to come down, and what they're going to do is sitting right out here is a receptor, a dendrite, right? And it's going to attach onto the dendrite, and it's going to stimulate this dendrite. And what's going to happen now is we are going to start getting a signal going towards the brain, which I'll talk about later. All right, so that's the way salt and sour are going to work. Now let's take a look, because these three are going to work the same also: sweet, umami, and bitter. So let's go ahead and erase this. All right, and now these are going to use a G protein coupled receptor. So let's go ahead, and now we're just going to draw another microvilli, right? And this is the tip. Now again, these work the same. I'm just going to use one example, but remember that we would never find these receptors. So first of all, on these, if we look at this, my sweet is going to have a receptor that's called T1R2/T1R3 receptor. Okay, so that's going to go with the sweet here. Okay, umami is going to use what we call a T1R1. So you see that's R1, that's R2, /T1R3 receptor. And then bitter is going to use what we call a T2R receptor. Okay, so again, we wouldn't find more than one of these type of receptors on this microvilli. So let's go ahead and let's start with the sweet. So let's use my blue. And what I'm going to do, this would actually be down over here, but I'm going to draw it over here. I'm going to have a 7 transmembrane receptor. So it's going to go 1, 2, 3, 4, 5, 6, 7. And again, this would actually be down more towards the tip of this, but just makes it easier to do it right here. Attached to this now, I am going to have a protein which we call gustatory. So there's my gustatory attached onto this, okay? Now attached to gustatory, I am going to have another molecule here, which we are going to call GDP. Okay, so that's GDP. I don't know how well you can see that. but this is GDP. And GDP inactivates this, or it's attached to this when it's inactivated. When a sugar, in this case, comes on, or if it's one of these receptors, if it's one of those, or if it's bitter and it comes on, what's going to happen is it's going to activate this, and it's going to activate gustatory. When this becomes activated, I get another molecule attached to this, which is GTP, right? And when GTP becomes activated, this leaves. So this is going to leave from that molecule. Now, what's going to happen now is my gustatory is going to go down here, and it's going to meet up with basically a molecule on here, which we're going to call phospholipase C, so PLC, right? Phospholipase C is then going to go on to a molecule which we call phosphatidylinositol biphosphate. That is too big of a word, so we're just going to call it PIP2, PIP2. So there's that. So now this comes down, it binds onto there, it's going to split this into two molecules. It's going to split it into diacetyl glycerol, which is not going to play that big of a role in this. The bigger one is going to be let's use black, it's going to split it into inositol triphosphate. Okay, it's going to split into inositol triphosphate. Now, inside of this, we are going to have the endoplasmic reticulum, also known as the ER. The ER is going to contain calcium. So here's calcium. It has a whole bunch of calcium in there. And then what's going to happen is when this comes over and binds onto a receptor, it's going to cause the release of this calcium. So now I'm going to get calcium coming out. Here comes my calcium. Okay? Now I get all this calcium that's going to be in here. And then what's going to happen is this calcium is going to come over to, you guessed it, another channel, and it's going to bind with this channel. And when it binds with this channel, what happens is I get sodium coming in. Here comes my sodium now. All right? So here comes my sodium coming in. And now the sodium comes in, and what's going to happen is when the sodium comes in, eventually we're going to get a graded action potential, right? We get a graded action potential, and what that's going to do is it is going to open up some receptors that are here on the cell membrane, okay? Now, if you recall, all cells make something called ATP. So we have ATP floating around in here. ATP is not inside vesicles. So when this depolarizes, or we get an action potential, ATP now is going to act like a neurotransmitter and leave the inside of the cell. And guess what's over in here? We are going to have a receptor, okay? And the ATP is going to activate this receptor. And then what's going to happen is we are going to get signals that are going to go towards the brain. So let's take a look now of what's going to happen now. So now we understand how these work. Remember, I am not going to have, this is only going to be sensitive for one of these tastes here, okay? So let's go on. All right, so now let's see what's going to happen next. So now what I'm going to do, and just for the sake of making this a little bit easier, well, let's go like this first. I am going to draw somebody's head. That's their nose. This is their eye, right? There's their eye. Okay, and then I'm going to draw an open mouth here, and then this is going to come back like this. All right, this is going to come back like this, and then there's their chin. So in the back here, we're going to have a few different structures. One of the things I'm going to have is the esophagus is going to be back here. So if you remember, the esophagus carries food to the stomach. So that's the esophagus. Then in front of the esophagus, I am going to have something called the trachea. And remember, the trachea has cartilage, so it stays open all the time. And on here, we also have something called the glottis. Okay, that's the epiglottis on there. And then we're going to have the back of the throat. So on these, we are going to have taste buds, right? We're going to have taste buds on the back of the throat, on the pharynx. We're going to have it on the epiglottis. We're going to have it on the esophagus. And then what's going to happen is these are going to come together, okay? And they are going to become part of the vagus nerve. Okay, so they're going to become part of the vagus nerve, which is cranial nerve number 10. And I always make my X's small so they look like a V. That's an X, okay, for cranial nerve number 10. So that's one thing that's going to happen. Now, like we already mentioned, we have our facial nerve, which is going to come over this way too. Okay, so here comes my facial nerve, which we said was cranial nerve number 7. That's my facial nerve. So then from the other side, we're going to have, and it's not from the other side, I'm just drawing this to make it a little easier to understand. We are going to have basically cranial nerve number 9. Okay, so there's cranial nerve number 9, my glossopharyngeal nerve. Okay, so there's my glossopharyngeal nerve. And then what's going to happen is all 3 of these are going to head to the same place. Okay, I'm going to make it look like they're coming together. They kind of do, but they're still separate. Okay, but just for the sake of right now, I'm just going to make it look like they all become one nerve. And then they're going to go up to an area called the medulla oblongata. And the medulla oblongata is at the base of the brainstem. And in the medulla oblongata, you have a nucleus. It's called the nucleus of the tractus solitarius. I'm just going to call it the solitary nucleus. Okay? And that's going to basically be in the medulla oblongata, and it's going to kind of go up into the pons. So this is going to be it here. So I should actually draw that there, right? So it's going to be in the medulla oblongata and in the pons. So let me draw my pons right here. So there's my pons. All right. Now, this is going to come up into here, and it's going to synapse. 2 things are going to happen when this synapses, okay? One, it's going to continue up into the brain, which I'm going to get to in just a minute. But I could have branches that are going to go to the salivary glands. They could go to the salivary glands, which are going to increase saliva, because remember, when you're eating, you want to increase your saliva, right? And the second thing is it could go to the stomach, and what it's going to do in the stomach is it's going to lead to an increase in stomach acid. But we're not worried about that right now. We're worried about this right here. So now this is going to also travel up, and it's going to go into the brain. So let me draw the brain here real quick. This is going to be my longitudinal fissure. Over here, I'm going to draw what we call the lateral sulcus, and over here is going to be my lateral sulcus. It's also known as the Sylvian fissure, right? And above this, on each side, I'm just going to draw it on one side, I am going to have something that we're going to call the insula. And the insula is deep in the brain. So there's the insula, and then inside the insula, I am going to have what we call the gustatory cortex. And if you recall, basically what cortexes do a lot of times, not always, is they're going to take this electrical impulse that's coming, and it's going to turn it into a taste sensation. All right? So now I have this 3rd order nerve. It's going to come up. Oh, I'm sorry, that's not the 3rd order nerve. But anyways, not yet. We're going to do that right now. So I'm going to draw my thalamus. You'd have your ventricle between there, right? And this is going to come up to the thalamus right here, and it's going to synapse again. Okay, so really quick, these are going to be my thalamus. This is my thalamus. Okay? And then what's going to happen is this is going to synapse again. And when it synapses, now I have my 3rd order nerve. It's going to go to here, and this is going to turn that electrical impulse into a taste and tell you what you taste. But now, do we like this taste or not? I don't know. So let's send this down to the hypothalamus, right? And in the hypothalamus, you are going to have your limbic system, hypothalamus, and in here is going to be my limbic system. So it's also going to go to the amygdala, it's going to go to the hippocampus, right? And what's going to happen is this is going to put emotions and feelings with the food. So do we like this food or not? And basically from there, now we know what we think of the food. So that's it for how we taste. Thank you so much for watching. If you liked the video, please hit the like and subscribe button, and we will catch you next time.